Gostaria de chamar aqui a minha companheira de mesa. Gisele, cadê você? Olha, vou iniciando
I
agradecendo muito
will
o convite.
start by thanking the invitation of Daniel and Carol. The event is very well organized, very interesting. And yesterday we had a great dinner. I danced a lot. I'm a little bit hoarse. I'm sorry, my voice is a little bit hoarse. I will start by inviting Delio, who has a degree in medicine at the Federal University of Minas Gerais. He had his master's in experimental biology at the Federal University of Rondônia, a doctorate in clinical research at Fiocruz. He is currently the researcher and clinical director of the Center for Tropical Diseases in Rondônia.
Pega esse, se você fizer o fixo do...
Som. Ok. O seu apontador é esse mesmo? Ah, tem. Ainda são bom dia, né? Quero começar
Good
agradecendo
morning.
ao Daniel e a Carol pelos convites, a organização como um todo. É um prazer estar aqui com vocês
Thank you
hoje.
very much to Danielle and Carol for the invitation and to the organizing committee in general. I will do my lecture in Portuguese.
Eu quero começar dando conselho para os jovens
I
pesquisadores.
would like to start with some piece of advice to the young researchers and say that the success, maybe what adds value to you as a malaria researcher, is pretty much the same thing that adds value to real estate when you are in the real estate market. The biggest value of the real estate is its location. So in my career, I have been very successful and I have been having some credit because of where I am located. I am in Sepeng, in Porto Velho, in the middle of the Amazon, in the bottom part of state of Amazonas, and in the border with Bolivia, in the region that there's a lot of malaria. People search for my services because I'm in a good location, and among these groups that talk to me, I had Siemens, which is an important company that they created a machine, an automatized blood test machine. So they had a diagnosis for malaria function. And I never had heard about that before, so maybe we could come up with a partnership. And they told me, but we don't have money. And I asked, what do you have then? And then they explained that the test uses a special algorithm that uses artificial intelligence. I used to teach in the university medical informatics, which is different than bioinformatics. So I really like informatics. I really like equipment. And this machine had a diagnosis for malaria. So I said, I'm going to I could pay to work with you now. So this is my disclaimer. This title is very vain when I say the state of the art, but actually I'm going to tell you my story in learning what this was, how to use this machine, and my curve of learning, how to use automated complete blood count to contribute to the malaria diagnosis. We have our famous gold standards. The microscopy continues to be very important because it is the morphological arbiter. But with the microscopy, if it's well done, we have the stages. We find alterations that we can only see when you find the parasites. We have 100% sensitivity evaluation in microscopy. But when you don't find the parasite, then you use qPCR, which is fantastic. Everybody knows really well to assess, especially the quantitative part. It is the entrance door to molecular assessments, to finding alterations. So these are both fantastic. These are references to us. So everything that we compare with these two methods. But these two methods, they have a filter, something complicated to them, which is the volume of work. If you work in the Amazon, if you work in the endemic area, we know that microscopy is a quick test. It is truly quick. It can be done in 20 to 30 minutes, maybe an hour. But when you have one patient, but when you have 10 patients at the same time and you have one microscopist to read it, maybe that could be a bottleneck. At CPEM, some days we have 5 positive patients, and some other days we have 30 positive patients. But the positives represent 10% of the ones that are tested. So you can do the math and see how long it takes. This is a bottleneck for microscopy when you have a very high volume. And PCR needs a greater structure, a larger equipment. It takes a little longer because you have to collect the sample to work. It is not something that is available to you in the hospital. We have the point of care. So we have maybe a quicker response that demands fewer experts to work, which are the quick tests. They have very interesting prices nowadays, and I'm going to speak about this at Gates' presentation tomorrow afternoon. We saw advantages and disadvantages, but there's another bottleneck in the molecular microscopic method. It only works when someone suspects that there is malaria. If there's no suspicions, if there's no diagnosis hypothesis, nobody asks for microscopy or a PCR or a rapid test. So we see an opportunity for this kind of blood count panel in some scenarios that will use a complete blood count in emergency services with a huge volume of people going through, and there are some febrile symptoms that are not that clear. The first thing that they do is a blood count. So thinking about malaria, since we have a blood count, in a febrile case like this, maybe we could test, maybe we could think about considering malaria, having the chance of doing this diagnosis in these cases that had no suspicion. So maybe there's the opportunity to have an equipment like this that can signal that there's possibly malaria there in regions where we didn't think about before.
And how do the tool work? I think I'm presenting a PDF file instead of my PPT, but that's okay. It could be a simple equipment that you could find in any emergency unit, any lab. It's automated. It could be simple like that, or it could be sequenced with several types of readings concerning several aspects of the disease. It could be monitored, as we see here, someone presenting this single tube. It could be completely automated with this rack here, and they are changing the tubes automatically, and it brings this kind of reading. It's a flow equipment. It's not so elegant, but it's bringing this morphologic distribution, light deviation, and it translates that because it reads some of the patterns and translates that into amount of red blood cells and white blood cells, and that's how it works with the body. But how the breeding is made and the detection? Most of the CDCs the completely automated machines, they do the first two ones here. It's a reading showing the amount of cells per destruction or consumption, a simpler one, or some are more complex, evaluating the morphology, the amount of platelets and red blood cells, or if there's a typical monocytes and neutrophils, and they're able to classify that to us. Some of the most modern ones has 5 to 6 parts, and sometimes it has light and laser, a different color for malaria, this violet laser flow, and it's able to identify with this dye that it's not a classic expensive marker as we have with cytometry, but it's able to identify the DNA and the RNA within the blood cell. But do we have DNA and RNA in red blood cells? And this is the explanation. And this is what it's going to find when it sheds the light. They're going to find DNA and RNA of the plasmodium in the red blood cell. A mature red blood cell shouldn't have it, and it's able to detect some of the equipments that have this laser, of course, but they are able to differentiate the infected RBCs from the non-infected RBCs. And they are able sometimes to identify between Vivex and falciparum. Another detail on how it's done. So evaluating platelets, 60% of malaria cases and they have thrombocytopenia. So it's a sign for the equipment. And identifying Vivex from falciparum because we have sometimes a different parasite load, the type of red blood cells. So the equipments are becoming more and more smart, and they are able to have different readings. And this is a study published in 2021. Someone was using one of these equipment in an initial way, showing how powerful this is. Talking not only about malaria, but when Siemens brought the proposal, they said we have tests for malaria and dengue fever. So that's going to be quite interesting. So I was studying and I saw some studies about that, not only those by Siemens. We have 324 cases and they collected samples of the patients with malaria, dengue fever, leptospirosis, typhoid fever, and rickettsial infections, and they had this automated blood test, and they were able to show that we could identify the differences. So this is a histogram, a type of graph. This test brings, and it's a normal control. And we here has malaria. We have changes in lymphocytes and monocytes and lymphocytes and monocytes, and the change of the histogram was enough to show that. And here we have a dengue fever alteration, and it's different from the normal one, the malaria one, and the typhoid fever that is we couldn't have this differential reading. So this is another chart that is present in the readings. That is a scatter plot that it's similar to what uh, the flow cytomic people sees. This is the control. B is malaria, C is dengue fever, and D is typhoid fever. That is more similar to the control, with not much difference. And this is the third way that is a bidimensional and three-dimensional vision of the control, malaria. So you really see the difference, and the dengue fever didn't present a difference compared to the other pathologies. So this is something that is able to deliver an advantage with some sensitivity. So what's the difference in terms of innovation?
automatizados,
brought by these
natural
automated
aos sistemas operadores
CVCs,
é que não estão usando
the new
só
generation.
uma
The difference is
estatística,
that they are not
mas também
using this
conceito de
fixed algorithm,
condições,
using
usando
some fixed
inteligência
positions.
artificial
They
para
are using
aprimorar o
AI
algoritmo, de
to
forma que ele
enhance
fique
the algorithm
adapto um
in a
pouco
way
aos
that it's
escopos
going to
especiais.
become
Então, ele
adaptable
consegue
and
fazer
able to
beneficiar todos esses dados, né,
differentiate.
que
So
entram
it's
na
able
plataforma
to
inteligente
bring
através
together
de
all the
alguma
data within the
suspeita
algorithm, and we could have a
alerta
clinical
para malária.
suspicious
Ninguém
suspicion,
esse algoritmo,
this
né, falando, nossa, essa
clinical
informação
alert
é
on
interessante.
malaria.
Não, a ideia tem sido
So
aqui é
the idea
criar
is
um
not to
radar
provide the diagnosis
possibilitado
and treat.
de
The
malária.
idea is to bring this alert on malaria
Então,
suspicion.
fazendo um resumo rápido,
Just to
nós temos um
summarize
CBC
quickly, so
base,
this
que
is the basic
tem uma
CBC
avaliação de algoritmo mais especificamente. A gente
without
tem
this
uma
specific
trombocitopenia, uma
algorithm
melhora junto, né?
identification.
A gente entender
So
que pode ser que
that
essa
could
doença
help us
prevaleça
understand
em
that
outra clínica
there's a
acompanhada.
malaria
Mas
if you have
esse
another
grupo
clinical analysis. But this group of
especiais,
CBCs, this
eles
other
têm, já, às vezes
equipment
tem um
pieces,
laser
they
diferente
have sometimes
para o delineamento, às vezes dá
violet
diferenciação
lasers
entre
trying
tipos e às
to
vezes
show the
ajuda nessa
DNA,
decisão.
bringing sometimes the
Hoje, na minha
types and the
satisfação, o
differences, and
aprimoramento tem ido
sometimes
muito bem
it only has
do algoritmo e isso
the
melhora bastante a
algorithm
sensibilidade.
enhancement that
E
will
o último
improve its
aqui, gente, esse
sensitivity.
último aqui
And the last
é
one here
incrível.
is
Né? Esse
incredible.
aqui eu trouxe um deles que chama
I
Sysmex
brought one of them called
XN30.
Sysmax
Ele tem uma
XN31.
parte dele específica para malária,
It has a
né? E ele
specific
usa
part only for
inteligências
malaria,
preditivas para
and
malária.
it's
Então,
using
ele dá um
uh,
resultado
specific
muito
malaria
melhor.
reagents. So this is
E
bringing
aí,
a
vou
much
começar
better
pelas, vamos
result.
falar do melhor, né?
And I'm
Vai
going
assim,
to
porque
talk
a gente,
about
né,
the best one.
escolher alguma coisa mais ou menos assim, né? Quero ver o
That's how
melhor, o
we
mais caro,
choose
aí tem
something.
esses números
I want
todos,
to
né?
see the
Para
best and the most expensive, and
começando
when
pelo
we don't have money, we go down in our choices.
né? Ele
So this
tem
is the Sysmax.
incríveis sensibilidades
It has
100%, mais de
this
99
incredible
e
sensitivity of
tem, como eu falei,
100%,
tem
a
alta sensitivity por mesmo
specificity of
específicas por dados,
99.46%,
laser específico
and
e tem reagente
they
específico
have
também, né?
laser-specific
Então eles
tools
conseguem
and
detectar uma sensibilidade
reagent-specific
perto do melhor
tools, and
microscopista.
it's
Então esse
better
aqui
than the
consegue
best
detectar e não detecta parasitas, detecta
microscos-
então células celulares infectadas
microscopic
por meio do
technician.
laser. Então tem uma
So
visão
they identify
ok, qualquer problema nele
the
custa
infected
150 mil
red
euros,
blood cells
né? E uma manutenção
per
super cara, mas
microliter,
fora completamente a
and
nossa capacidade de trabalhar.
it costs a
Aqui
lot more,
um exemplo do
30,800
rodamento
euros,
dos
but
lasers, né?
have
Igual esse
annual
aqui, uma
maintenance.
situação
So
fácil de
here we
fazer,
have
fácil de
this
qualquer outro
fake
lá
positive in
lidar como
this,
brigadeiro
and
e
any other
conseguir
are
fazer muito bem
telling
feito.
them as true, and
Mas
it
não é só
was
isso.
able
Ele é
to
muito
tell the
incrível.
difference. But
Ele
this is
esse aqui
not
chama
our reality. And this other
Mind
group,
Ray, ele
which is called
exatamente
Mind
isso
Ray,
que eu só
it's able
só um
to
tipo de
show
laser e ele consegue identificar
to
então
shed the
RNA,
laser,
bom para encontrar
and it's
troponídeos,
able to
né? Então
identify DNA
basicamente
and
ele
RNA.
tem
It's good to
muitos neurônios
detect
circulando e até
trophozoites and
schizons. Então ele é ótimo para
schizons,
subambientes
and it's
especificados,
great for
é ótimo,
Vivix.
valor preditivo negativo é
Sensitivity is
excelente,
great,
mas
specificity is next
falciparum,
to
o schizon
100%.
está lá
But
guardadinho
for
na microcirculação
falciparum,
e
the
passa rápido
schizontis,
a sensibilidade dele.
it's
Então,
saved in the
funciona muito bem. Esse aqui
microcirculation,
também
and
só para
so the
você ver,
sensitivity
fantástico.
is
Tem a
not so
visão
good. If it's only
do
Vivix, it's
troponídeo. Então,
fantastic.
e aí
But
a gente
if you
tem,
have both
então, esse
distributions, it's
trabalho para a gente fazer
not
um
good
leque
enough.
teórico, que
And
é
we had
a
this
rede
work in
genérica para malária
Porto
530. Ele
with
está na
this
linha também meio ali, ele não é mas tem
M530. That is a
CBC
middle
até
line,
o
a
mais
normal CBC, but
muito mais
with
potente para poder
this
avaliar
very
esse
powerful
tipo de problema.
algorithm
Então,
to
Porto Velho, como eu
identify.
falei, nós estamos bem
And
aqui na região amazônica,
Portofelho
então ele é
is in the
tradicional.
Amazon
Então, para
region,
quem
and
quer
we attract
brincar, que
that.
consegue nessa área,
So
esse equipamento consegue ter um
the
diagnóstico interessante, a gente montou
those
um estudo
patients
aqui
with
com
fever,
pacientes
could we have a
filhos
good
que chegam
diagnosis? We
também.
had
A
the
gente
study
conseguiu fazer
designed with
um
500
diagnóstico
patients with
antes de
fever.
fazer malária, a gente
And
observava eles
before the
e
malaria
observava no
exam,
exame de
we
sangue, e aí ele ia
approached them
fazer
and we collected blood, and
o
then
PCR, a
we did
microscopia,
the
e
PCR, the
a
microscopy, and
tomografia
the
com
reading
laser. E a gente
with the
fazia isso
CBC.
para os cegos,
And we
né?
were
Então,
able
os caras
to do
cegos
that with
que
double
não sabiam que
blind.
estava
Those
fazendo
reading
o
didn't
exame até
know
chegar na hora do
what
exame.
was being
A
done
gente tem aqui
until the
diversos,
analysis.
contei casos
I'm
demais.
I'm running because
Então,
I'm out of
a gente
time,
teve em
but
todo
in
498
total we had
amostras para análise.
498
E a
samples to work.
gente acabou tendo
and we ended up having 158 positive cases and 340 negative to PCR. And analyzing, we have this um, accuracy, the sensitivity of 87.34, the capacity of identifying the true infected people, the truly infected people. The specificity was 79, the negative predictive value was 93.10, and a positive predictive value that was low. It's a clinical important result, but when I come to with this negative test in my hands, if it's negative, how much could I trust it that it's negative? So 93%. But if it's positive, only 66%. So I should do a different test. This then we could refer the patient to microscopy, a rapid test. So in summary, that's what we have. A conventional is exam that is automated. So it's here with the sensitivity of the common hemogram against 70%, but with 56 of co. And then the second line, we have laser, and we have the EMA technique that we are using in Porto Velho with 99% of sensitivity, but for falciparum is not so good. That was for Vivex, and we couldn't evaluate falciparum because we only had Vivex, and we had this global evaluation. And the CIMEX, the wonderful one, that is very interesting, but it's very costly. Concerning some critics, we have the sensitivity that is not so good for species differentiation. We have a lot of false positives. We should improve that in this intermediary uh, line, and the very good ones are very expensive. So we have a lot of data, and these equipment are able to survey the data, but the human eye is not able to evaluate. We must work with intelligent algorithm empowered by artificial intelligence, and the change shouldn't maybe happen in the hardware, but in the software. Software is what you call the app. We should change that. So Siemens was lucky enough to work with Gates Foundation, and they presented the data from Porto Velho, and Gates said that they wanted to develop the algorithm even more. So the Porto Velho data is now being improved with their algorithm, and Gates didn't waste their time because they paid us if they develop this agnostic equipment that is working not only for Siemens but in any CBC. You don't have to change the parts, only the software. It's an update that is going to identify malaria. It's a very interesting promise, and our role is to understand where this type of equipment could be useful. Maybe in places where we have this incidental triage screening, and it could flag us that it could be an endemic place. So maybe all of the fever cases we should have the the analysis in the lab, but this is not happening. Many are not doing that, and we are only observing cases much later. So some other cases, and this is the last slide, and what's happening is that we got this information from extra Amazon region. We have 99% of malaria in the Amazon, but the mortality in the area is 50 times greater outside of the Amazon, because the doctor outside of the Amazon region, they do not suspect malaria. That's why they do not ask for the specific exams. So that's why we should maybe use this type of equipment. But I'd like to thank the lab team the clinical studies. Michelle is the leader. Our team from CETEM and Attack team is who lent us the equipment. And André Daya is in the picture. He was visiting us, and I consider him part of the team because ever since we were putting the team together, they helped us with the clinical trials. So it's a pleasure to work with those who want to work with us, and that's it. Thank you. Thank you,
Obrigada, Délio, pela sua
Dalio,
apresentação.
for your presentation.
Bom dia a
Good
todos os
morning
presentes.
to everyone. And moving on with our roundtable, I'd like to call Alexandre Dias Tavares Costa. He is graduated in biological science in the State University of Campinas. He has his doctorate from the Federal University of Campinas and a postdoc in Portland State University, where he also occupied the position of research assistant professor during the period from 2005 to 2007. Among some of the roles he occupied in the Molecular Institute in Paraná, IBMP, we highlight the technical management of research projects and development of new products. Nowadays, he is a researcher of Oswaldo Cruz Foundation at Fiocruz in Paraná in nanobiotechnology and prototyping, being experienced in biochemistry, molecular biology, and physiology, highlighting innovation and technical development. Thank you so much.
Bom dia, pessoal.
Good morning, everyone.
Bom dia a todos.
Good,
Carol e Daniel,
Carol and Danielle, thank you so much for inviting me. The Congress is really excellent, very, very interesting, and I hope to bring to you some news on what we've been developing. We are finalizing this validation work
trabalho de validação de teste de PCR
of
baseado
this
em PCR em
uh,
campo.
qPCR-based test in the field. So basically, we've been working for 10 years with Delio's group, developing since the beginning this new reaction
Para chegar teste de PCR
to
factível em
get
humano.
to this feasible PCR test in the
O
field.
diagnóstico é pilar
And
essencial.
diagnostics is an essential pillar. I don't have to tell you about that.
Testes que sejam mais
And we
objetivos, né?
must have less subjective
análise de
tests
teste
with
facilita
more
também quem está
objective
fazendo
tests because
a
it's
definição
easier
da
for
resposta
those
clínica.
who are making the clinical decision.
Cura, né, para
In
eliminação
terms
da
of
malária, um
cure
ponto
for
fundamental.
malaria elimination, an important point that was mentioned here very much was the diagnosis of asymptomatic patients.
Isso
And
não é nenhuma novidade, né?
this is not
Mas
new,
a grande
but
dificuldade, então, é
what's hard for
atração
us
dos
is to
investimentos. A gente
attract,
tem essa grande dificuldade, não só no Brasil, mas no mundo inteiro, de
not
atrair
only in Brazil,
profissionais para uma
but
profissão que
all
não é
around the
tecnológica.
world,
Eles
attract
entendem que a
professionals to this
tecnologia
non-technological professionals because unfortunately the technology
né? Então a
is
gente não quer
coming
mais trabalhar com uma coisa que não
for
seja
a
tecnológica
simple
materna.
counter and no
A
one
gente
is
tem
willing
que
to work with something that is
lidar
not
com
technological. It's a pity, but we have
a
to
vida.
deal with life.
Então, a pergunta que sempre que ficou colocando com o
So the
tempo de
question that
biologia
has
molecular
always been around during my time at the Institute of Biotechnology of Paraná was that we were developing new molecular tests, and we were wondering, are these tests going to be used only in the central laboratories of the states, only where there's big infrastructure?
tem, né? A gente está aí desenvolvendo
We are here developing the national industry. This is good, but effectively, this is not solving the problem. So the question that we asked ourselves was, is it possible to have a real-time PCR test that we could take to the field? And as a bonus question, after talking to the personnel of the ministry for a while, was, is it possible to detect the symptomatic patients, the close contacts of the symptomatic patients. In this region, we always see that person that does not catch malaria. Oh, I never catch malaria. So can we show these people that actually they do catch malaria? So whenever I show our work, I show the portable PCR equipment that we use, and everybody says, that's beautiful. Oh, that's wonderful. I just wanted to make very clear to you that the portable PCR equipment is not new. It is the technological part that draws the attention, but it's nothing new. After the pandemic, we had dozens of portable PCR equipment, real PCR that are portable and that could be taken to the field. So what is missing? What is missing is a process of DNA extraction that is simplified enough for you to execute in the field, but that does not lose a lot of sensitivity and specificity so that you can bring a benefit to the diagnosis of malaria in the field. I don't want to have to bring the patient back to the laboratory. I want to diagnose the patient in their house. So initially we worked with tuberculosis, and now we have validated the reaction for we started with tuberculosis. As I said, it is a very simple process of collecting the sample, measuring the volume, adding solution 1. It is in the paper written there. What is the solution 01? But you apply the mixture onto the elute microcarb paper. The dozens of different types of filters that exist, probably you know about them. We punch a 6 millimeter disc, we add the buffer, and you have there the extracted DNA solution. It is a crude solution, but it already allows us to run PCR in real time. So in 4 hours, in 3 to 4 hours, we can collect the sample, extract the DNA, and run the PCR next to the patient in the field in any situation. None of these equipment here, none of these steps are something that demands big equipment or more robust infrastructure. This filter paper, yeah, usually the blood that is collected for malaria is collected in a classic, ordinary filter paper. This filter paper has detergent embedded in it. It is sold by Wattman. The difference is that there is detergent embedded. So when we do the first manipulation of the blood before we add it to the filter, that brings us a fundamental difference for the procedure of use of classic use of filter paper that people apply. Here what we do is to treat with protease the solution. So when I'm adding the suspension of blood cells to the filter paper, they're pretty much all denaturated, and the detergent that is found in this paper helps releasing DNA. So with that, I did not do any study specifically on that, but this difference for me is paramount to the fact that we were able to effectively use the DNA extracted from the filter paper in the field. Everybody who knows PCR will tell me you're in the field, so how did you work with the reagents of a PCR? They are thermosensitive. Usually this is the standard procedure. You extract your DNA, you prepare the master mix, you add it to the dish, you add it to the plate, and add extracted DNA. And right now, in this moment here, is where most of the mistakes happen, doing the calculation and adding it to the plate, the 96 plates. We can facilitate all of this process, doing all of this in the manufacturing process. So we managed to mix the PCR reagents with the gelifying and stabilizing reagents. We apply vacuum to that solution and we form a gel that is capable to of maintaining the activity of the enzymes and the components of the PCR up to 1 week if we keep it on room temperature, and up to 6 months if we put it in the fridge. Just a comment, room temperature is the temperature of Curitiba, okay? It's the temperature of the south of Brazil. Let's not compare it to the temperature of Pará in the north. So basically, for transportation, we can think about sending the agents in room temperature, but basically we would have to always keep those reagents in a fridge temperature. Thinking about what we have in Brazil with SUS, if I want to remove the test from the laboratory, I don't need a freezer anymore, I just need a fridge now. A fridge that is found basically in all of the healthcare facilities because they have to keep insulin and some vaccines. So family health clinics, they do have a fridge. So we have all of the conditions that we need to take it to the field. We have a simplified procedure. We have a new way to store the reagents. And as I told you, it all fits in a DoorDash delivery backpack, and with that we can go anywhere. We can put it on our back and go anywhere. The equipment that are there will not move around because it's tightly packed. So here we can see the backpack and at inside the pickup truck because we went with the health agents to the farms, and over here in a religious community.
Tratamento de drogas, né?
It's a drug addiction facility, religious facility for people who are isolated 20, 30 kilometers inside the woods so that they can't leave. There's no fence. There's just 20 kilometers of wood to the next city. And in these villages, these small communities, there's 30 to 40 people, and it is a transmission focus. So we managed to go there and use the infrastructure that they had available to do our tests. We went to the region of Porto Velho, in the rural area of Porto Velho, and the residential area of Porto Velho, also to the central lab. We also did a passive search in the region of Vista Alegre do Abunã, where we had only a healthcare facility for a family clinic. And in a second area where we carried out the testing on site in the area of the Leiteira Basin. We would go to the people's farms and or the community houses, and we would meet people there and do the testing there. So we would spend 2 or 3 days at the site running tests. In terms of results, what we had was the collection of 399 samples. We did microscopy on them, and we compared them to the lab test. We also took the blood to the test to use commercial kits. If we add these samples, both symptomatic and asymptomatic, we can see that there's a difference in the commercial kit and the microscopy, and our test is not that much better than microscopy. Right now, at that moment, we had 130 samples, and we talked to the ministry, and they said, for symptomatic patients, we can find them. We cannot find the asymptomatic cases. So over here, we have the general figures. There is a different detection. The commercial kit still detects more than microscopy, but the portable test can detect samples that were not detected by microscopy. So here we can segregate by falciparum, by VVAX, and also mixed infection, which is a little bit more difficult to identify with microscopy. After talking to the ministry, the difference is that we segregated our data between symptomatic and asymptomatic cases. When we look here, we see that truly there's not a lot of difference. So microscopy is really good to detect the symptomatic ones. The commercial test cannot find so many more cases, a lot more cases than microscopy. And the big difference appears when we look at the asymptomatic cases. Microscopy almost cannot detect asymptomatic cases. Commercial tests finds more cases, including some mixed infections, and the portable assay can detect approximately the double of mixed infections in asymptomatic cases and half of the commercial results for falciparum. If you like figures, in asymptomatic cases, we basically have very similar numbers for the, for the symptomatic cases, we have similar numbers for the three techniques, but when we look at the asymptomatic cases, we have a considerable difference. So with the test, we can detect the double of positive patients. The numbers depend on who you're going to consider, what you're going to consider as your golden standard testing. The numbers are a little bit better when we consider the symptomatic cases, of course. That is where we have the biggest correlation. And when we have the asymptomatic cases, we have a difference of sensitivity and specificity. The big issue that I would like to show you is not if the test is perfect or as good as a commercial laboratory test. We know that this is never going to be the same. What we want to have is a product that is capable to be taken to the field and provide an extra tool for us to find those asymptomatic cases. It's not going to solve our problems, but it's going to allow the field agents to make a more informed decision. Right now, we are at the moment of fine-tuning of the test in the pilot scale, and we're now demonstrating the prototype in an operational environment with reagents manufactured by a company. Everything that I showed you so far were reagents that were manufactured and produced inside the laboratory with all of the uncertainty that the laboratory has. We work with the best practices in our lab. Fiocruz has a lot of different criteria for their labs, but we still have the uncertainty of a research laboratory. The next step, if you don't know, is to have a contract with the company and have the company manufacture all of this and go back to the field to show the results with the company's reagents. So we're going now to the region of Rodrigues Alves. It's an elimination region. One of the situations that we want to understand in Mariana de Cima, for example, for 2 years, we have had no positive cases. Last month, we had a 2-year-old that tested positive. So where did this infection come from? There was no one new arriving in the community with symptoms. So someone asymptomatic arrived in the community and started this transmission. So what we want to have is give the healthcare agents a tool to find this person and keep this area free of infections. So the conclusions that we reached is, yes, we do have a real-time PCR test that is not perfect, but it's capable of detecting VVAX and falciparum in real-life field conditions. It has accuracy similar to microscopy in symptomatic patients, but we trust our microscopists. And it has a accuracy and positivity that is twice higher than microscopy in asymptomatic patients. the question is, can we use this tool to eliminate this community transmission, as the case of Mariana de Cima, as I mentioned to you? This is our idea. So in addition to this, we can also suggest the use, that little box, that little DoorDash delivery backpack. It doesn't have to be only in each family clinic. It could be in the main city. And if there is an inexplicable outbreak in a certain location, the agents go there with the molecular test in their backpacks, and they can run the test anywhere. Here, for example, we were in a religious community, also running tests at their ballroom, at their party room. Another suggestion that we can think about is having this equipment at the border, where you have where you have interesting movements. We're not reinventing the wheel. Once again, the idea of taking tests already exists in many parts of the world. We have bicycles or animals carrying the tests where you need to go, and our test fits this type of solution. And to echo Professor Bento, who spoke earlier, it is important for us to think about the territorialization, looking at each environment and each specific situation and having several tools and using the tools specific to each one of these situations. I would like to thank everyone who helped, especially Delio and Paulo Fontoura, and our students and the financial and institutional support that we received. Thank you very much.
Agora nós vamos chamar a
Now we
próxima.
are going to invite the next speaker, Natalia Nogueira Chama Siqueira. Natalia is a biomedic with master's and doctorate in biology of parasites and diseases at the University of Pará. She is a technologist in research and biomedical investigation in the laboratory of malaria of the Evandro Chagas Institute, and she has actions of surveillance in health in the area of parasites since 2009, with an emphasis in malaria.
Bom dia a todos.
Good
Agradeço
morning.
a organização do evento.
I'd like to thank the organizing committee for inviting me. It's a great opportunity to be here in this discussion forum, being able to bring our reality from the Amazon in this extra scenario. It's very encouraging to participate in these events. I'll try to be objective because we've had excellent presentations, so sometimes we could overlap a little bit. I'll try to be brief and objective. My focus here, when we talk about, is that we are going to talk about point of care testing for malaria diagnosis, but I don't want to talk about a single tool or methodology. I want to talk about strategy in terms of point of care, aiming at accessibility,
desenvolvimento tecnológico e,
the,
sobretudo, a tomada de decisão.
the access technology in decision making.
Que os testes point of care importam no
Why
cenário de eliminação?
POC matters for elimination. As it was said before, we all know that diagnostic is a critical element for eliminating malaria, but we must remember that we have two epidemiological factors that are time and access. And many times we must give up the analytical perfectionism to enhance accessibility and this diagnosis possibility. And the point of care test,
Estou com algum delay aqui no. Está dando para ver. O teste point of care
the
ele
point
vem
of
como
care
uma
test
estratégia
is a
para
strategic entry
atender a
point
essa demanda de
for
tempo e acessibilidade.
this time and
Uma
accessibility
atenção aqui para a gente é
aspect.
que os testes point of care eles não têm
The
exatamente
point
o
of care
intuito de
tests,
simplificar o
they don't
diagnóstico,
aim at
sim
simplifying
assegurar
the
a
diagnosis, but they want
sustentabilidade
to
dessa estratégia
secure
em
the
cenários
strategy
desafiadores.
sustainability in challenging
Por
scenarios.
que a Amazônia ela é um cenário tão particular, né?
And why is the Amazon a strategic setting?
Que reúne características
It's a region
como
with
a
different
alta
features,
mobilidade
such as
populacional, destaque
highly
muito
mobile
instável e
populations with
anticontínuo,
unstable and
serviços de
heterogeneous
saúde que eles
transmission.
são intermitentes,
The
a gente convive
We have
regularmente com
intermittent health
desvios econômicos
services,
nesse
and
cenário,
we have
muito crítico, porém real,
the health
além
services
das
being
longas esperanças
discontinued
e
many
possibilidades
times. This
muito
is critical and it's real. We have long distances between care and
muito
reference
desafiador.
labs, and
Mas
these are
dentro
different
desses
challenges.
desafios,
But aware of the
temos que
challenges, what could
buscar
we do?
a flexibilidade
We
não como
could
uma
try to
opção,
be
mas nós
flexible,
precisamos
and
partir dela,
flexibility is
né?
not
Nós
optional.
precisamos ser resilientes
We
nesse
must be
sentido e
resilient. We
nos
must
adaptar a esses desafios
adapt to
para
the
não simplesmente
challenges.
ficar nos lamentando por eles. Então, os testes
So
point of care, mais uma vez, eles
the
vêm
point of care
ao
tests,
encontro dessa necessidade como uma
they
estratégia muito
meet
valiosa.
the needs as
Eles
a very
podem
valuable
promover
strategy. They are
o
able to
tratamento
promote
imediato, que também é
immediate
crítico para a
treatment.
eliminação da malária.
This is critical
Eles
for
promovem
malaria
a
elimination. They are able to
estimativa e
promote the
resposta
active and
rápidas.
reactive case
Promovem
detection.
uma resposta rápida, que é
They
um fator
also
também
promote
fundamental,
this rapid response,
em
which is also a
eventos
key factor
onde a transmissão está
in
ocorrendo.
events where the
Eles
transmission is
também
happening.
permitem
They
a
also
sustentabilidade
enable
do acesso em áreas
the
remotas,
sustained
em
access in
cenários que
remote and
flutuam.
non-stationary
A gente vê de um
settings,
jeito,
because
pode ser
sometimes
de
we
outro.
see
Na
the
Amazônia,
scenario in
a gente
different
tem áreas que
ways.
passam 6 meses
And in the
alagadas, 6 meses
Amazon, we
em
have
seca,
scenarios that
e o
sometimes
acesso que é
spend
com
6 months
complexos, não existe
in
rede de
floods and
barreiras. E
six
dentro da
months
Amazônia nós
uh,
temos muita
in
diversidade. Então nós
dry
precisamos
periods. And in the
conduzir
Amazon, we have a
reflexões
lot of diversity.
como
We
Alexandre
must
falou,
understand
para
the
que a gente possa se
peculiarities, as
adaptar a eles.
Alexandre has mentioned, so that we can adapt.
Para construir essa estratégia
To
diagnóstica
build this
sistemática,
diagnostic
é
strategy in a
preciso
systematic way,
ver que o diagnóstico da malária ele não
we
deve
must take into
partir
account that
somente
the
de uma
malaria
tecnologia.
diagnosis
Nós
shouldn't use a
devemos
single
combinar
technology. We
recursos,
should combine
metodologias
different
para que nós
technologies and methodologies
possamos então
so that we
alcançar
are
a
able
questão da
to
chamada eliminação
achieve
nesse ponto
our
mais desafiador ainda, que é depois,
elimination
né,
and keep it later
diante
on.
dos desafios que estão postos.
In face of the
O
challenges
teste point of care
we
ele
have
promove
to
rapidez, cobertura e
to
essa
endure.
pronto,
So
pronto
this
tratamento também.
rapid
À medida
test
que, além
provides this ready
das
treatment
referências
with this
que se podem lhe
fast
dar, nós temos
results.
pesquisas aqui no local, né? O
Microscopy
ponto mais
is a
perigoso.
clinical reference
E as ferramentas
with
de
this species
tecnologia, elas já
confirmation.
trazem
And
suas
the molecular
características de
tools, they bring their
parametricidade
features
e
of
audível,
increasing
de forma mais
sensitivity,
sensível também à vigilância,
guaranteeing
em
surveillance in a
especialmente
more
no ambiente
sensitive
de
way,
eliminação.
especially
Então,
in
quando nós
elimination
combinamos
settings.
essas
So when we
características, nós
combine these
conseguimos
tools, we
avançar
are able
nos
to advance
objetivos de
in
eliminação.
our elimination
Em
objectives.
relação ao Plasmodium vivax,
Concerning
muito mais do que uma
Plasmodium
frustração,
vivax,
esse
much
desafio, e eu sei que
we
todos
have
nós
much more than a
amamos a
frustration.
ciência, amamos a saúde
We
pública,
do love
somos movidos a
science and
testes e como
healthcare
superá-los.
and
Então, o Vivax
public
ele faz
healthcare,
parte
and
disso,
we
né?
are driven by
E
challenges. And
os
Vivax
desafios
is performing
precisam ser
this
abordados
role
também
because
de formas
the challenges
peculiares.
must be approached
Mas
in a
além do que
peculiar
também
way.
sabemos, as
And
densidades
we
parasitárias
know
na
that
Amazônia
we
costumam ser
have
baixas,
parasite
elas mais
densities
prevalentes,
in the Amazon which are
geograficamente.
low.
É comum
We
ver
have the more
densidades
prevalent
parasitárias abaixo de 100
parasites. It's
parasitas por
common
micrograma.
to see less
E
than
é
100
muito
parasites per
difícil, num
leader, and this is very hard in this very heavy routine of microscopy. But the PDRs, which are very important, uh, they they are sometimes we are not able to use them, but we must overcome that. And we have a higher proportion of subpatent infections and the presence of reservoirs that go beyond routine detection. So POC ensures access the molecular tools with their peculiarity, especially in this context of sensitivity, uh, can uh, allied to POC, they could provide support to elimination. So this robust healthcare system must deal with risk management, not understanding that the risks should be eliminated. We know that malaria is not only a health problem, so we must have this holistic approach to treat different situations with this cross-cutting mindset that encompasses cultural aspects, political aspects, social aspects from different natures. And since we work with that, we must be very attentive. We must listen to the communities. We must go to the communities and experience their way of life. We are in a privileged place to work with the disease, but yet we must have the sensitivity to go towards those who are in the community. Maybe a riverside hub that could do the tests and so that we can assimilate and propose the right strategies for different realities. This way, we are able to promote the integration of surveillance with complementary diagnostic tools for challenging settings and evaluating data in a more and more efficient way that would drive us to right making. So I talked about all the aspects here, and where do we see the POC impact in challenging scenarios? So the topic here is the Amazon, but this is not only happening in the Amazon. You know that we have challenges in the extra Amazon settings, the lack of epidemiological knowledge, and we need different tools for different scenarios. But POC for the Amazon, it aims at solving some problems. It's the first contact of the patient with the public health system. I'm able to go to this indigenous village, as our colleague did, to help the workers there who were facing critical conditions, trying to promote diagnosis in this setting. And the TDR also helped us a lot, right, Alex? And also with outbreak containment as an active and reactive response in the surveillance context, and with residual transmission and reintroduction risk in very difficult contexts. The POC tests are suffering this evolution, as I've mentioned before. The intention is to remove subjectivity, but also improve the quality of the interpretation. And how do we do that? With digital integration, providing support to areas where the patient is, and integrating more and more the field to the laboratory. And the intention of the POC tests is not to eliminate the lab, but to stretch the lab's arms so that we can reach the reality. Concerning the objectives, we have many technologies being developed. Some of them were presented here, and I'm going to mention some more. Besides the simplified molecular platforms, we also have the association of
microscopia, promovendo
AI
assim
to
tecnologia
microscopy,
também.
promoting
Além
uh, tele
de ferramentas multimodais
diagnosis.
que
And we
também
also have
podem
multiplex
adaptar esse formato de
testing that could
toque-toque,
adapt
que são
to
muito
POC
úteis, por
tests, and
exemplo,
they
na
are very
identificação de
useful
síndromes febris.
uh, to different different different
Nós somos do
fevers.
Instituto Evandro Chagas, do
And we are
Laboratório da
from
Malaria,
Evandro Chagas
e lá
Institute in
nas
the malaria lab.
linhas que
And
nós trabalhamos
among the
também
lines we
no
work with, we
desenvolvimento
also
de
work with the
protocolos de
development
diagnóstico,
of
métodos diagnósticos.
diagnostic
E todos
protocols
eles são
and
pensados,
diagnostic
desenhados
methods,
nesse
and all of them
sentido
are
de
designed
visar a necessidade real
aiming
dos
at
usuários da Amazônia, já que nós estamos lá. Nós temos
caring
mais ainda
for
um
the
diferencial,
reality in the
é
Amazon.
integrar essa
And we want
vigilância
to integrate this surveillance
integrado à vigilância e ao
to
cuidado, ao
care,
serviço, e focar na escalabilidade
and we want
e
to
também
focus
no
on
suporte
scalability
técnico.
and decision support.
Aqui são dois técnicos do nosso grupo,
We have
e
two
um pouco mais
technicians from our
das
group and
ações
some
no
of the
estado do
field
Pará.
actions we have as a routine.
Eu trouxe aqui muito brevemente, eu vou fazer duas menções a dois desses projetos
Very briefly,
relacionados
I'm
com
going to mention
desenvolvimento
two
febris.
projects
O primeiro
concerning
deles,
the
desenvolvido com a Universidade
diagnosis
Federal do Mato
development.
Grosso
The
do
first
Sul,
one developed with
envolve
the
a
Federal
despaconga,
University of Mato Grosso
no
do Sul
sentido de
and
fazer,
is
promover o
engaging
diagnóstico de triagem
this
a partir
phot-
desta tecnologia.
photomic, photomic
Como que
technology
funciona?
for
Está vermelho aqui, mas isso é
screening.
um soro, tá?
It's in red here, but this is a
E a gente
serum.
aplica cerca de 20
And we
mililitros
applied
de soro
20
no
microliters of serum in the
Ih, gente, eu não estou conseguindo falar agora.
spectroscopy.
Espectroscopia, desculpa, que
In the
utiliza duas
spectroscopy
metodologias.
tool that is using two methodologies,
Que tem
FTI,
uma abordagem mais
that has this
qualitativa,
qualitative
associada
approach
ao
associated
NIR,
to
que tem uma
NIR,
abordagem
which has a quantitative
Então,
approach.
a partir dessa
So
análise
from this analysis,
de biomoléculas, proteínas
this
e lipídios, principalmente,
biomolecular
é gerada então uma
analysis
sintomatologia química daquela
with
perturbação
this
do
with the
agente naquele
profiles of
corte.
proteins, lipids, and
E
functional
essas
groups in the
são
serum,
testados
we are
diferentes
able to
padrões que
test
são
different
ensinados
standards.
através da inteligência
So we,
artificial para a inteligência
so the
artificial.
agent
O padrão que
teaches
seria
artificial intelligence
conversacional
the
e
standard
os
that is
padrões
considered
de
normal and
patológicos, né? E
pathological
é
standards.
possível que a
And
gente
then it's
faça isso
possible
de
that
forma
we are able
multiplex.
to do that
O
in a
desenvolvimento
multiplex
dessa
way
ferramenta, ele
with the
tem sido feito
development
não
of this
somente na
tool,
malária, mas é uma ferramenta multiplex para a
with
diferenciação da
this
malária,
multiplex to
leishmaniose,
differ
dengue,
from malaria,
zika, chikungunya,
leishmaniose, dengue
doenças
fever,
chagas
chikungunya
e
fever,
tuberculose.
Chagas disease, and TB, if I'm not
E no sentido de triar e fazer essa
and we
diferenciação
screen
com o
and
auxílio da inteligência artificial.
we differ them with the aid
Dessa
of
forma,
artificial
nós
intelligence.
conseguimos,
This
já
way,
estamos
we are
né, já
able
passando pela prova de conceito, mas os resultados
to
já são
have
bastante
the
promissores,
proof of
e assim nós
concept,
temos
but we
então
have
uma triagem
potential
rápida,
results so that we have
custo
this rapid
operacional
triage with
viável,
this
que
viable
assemelha a uma
cost that is
microscopia,
similar
e
to
também temos a
microscopy. And we
portabilidade.
also have
Então,
portability.
repito, a gente ainda está no
We are
momento de
still in
validação,
a moment
mas
of
a gente já tem
validation,
dados,
but we
já
have
tem
data
achados, e alguns deles
and
eu
we
não vou
have
ter tempo de
findings.
apresentar aqui
I
nessa
won't have
fala,
the
mas nós
opportunity
temos
to
duas
present
colegas da Universidade
some of
Federal
them,
do Mato
but
Grosso do
we
Sul,
have
que
two
são
colleagues from the Federal
a
University of Mato Grosso do
Yasemin, que
Sul,
apresentou ontem,
Yasemin,
e a
who
Marissa,
presented
que são
yesterday,
duas
and
outras
Marisa,
posteres de
and they are
diagnóstico
both
e
with
mais
their
detalhes sobre as triagens
posters
podem ser vistos
bringing more
com a nossa
details
equipe.
about the findings.
O outro projeto que a gente
The
está
other
também
project
trabalhando,
we are
junto com os
working
biomanguinhos,
with
também no
Biomanguinhos is
desenvolvimento de um
also in the
QPCR
development of
para
this
tipagem de malária,
qPCR
point
for
of care. E a
malaria
gente trabalha em
typing at the point of care,
dois workflows.
and we work with
Com
two workflows.
extração
One with
através
the
de vídeos magnéticos e
extraction
outro sem uma
using
extração prévia.
magnetic
Então,
beads,
a
and
gente
the
está
other
falando
with
aqui
no
agora de
previous
conceito,
extraction. So we
e
are also
hoje
in this stage
a
of
gente já
proof of
tem visto
concept, but
também bastante
what
interessante, a
we've
gente espera
been
na
able
próxima
to
reunião
see that is quite interesting is that we hope that we have consolidate data in the next meeting so that we can share with you. But another detail is that we use in this prototype uh, a chip that is preloaded with primers and probes, and it's all configured not only to ensure sensitivity and specificity, but also to remove subjectiveness from the analysis, enabling the work in remote areas. Last, I'd like to bring this reflection to all of us that in the Amazon, the malaria elimination will not be achieved by a single technology. I'm very happy to be here with my peers, being able to enable possibilities so that we can strengthen the different strategies, contributing with public health care, bringing new possibility to improve the health care and the situation of vulnerable populations, achieving the objectives of elimination. Thank you so much.
Obrigada a todos.
Thank
Tivemos
you,
uma mesa muito
all of you. We had a very
com
interesting
Zélia, Alexandre
table
e Natália.
with Delio,
Agora eu vou
Alexandre, and
abrir para
Natalia. And
perguntas.
now I'm going to
Por
open
favor,
for
quem quiser se
questions.
aproxima do
If
microfone
you
que está
want to
aqui.
ask questions, please come closer to the mic.
Já tem uma fila.
We have a queue here.
Bom dia, muito bom. Foi
Good
uma
morning. It was great, an excellent
para qualificar os
roundtable
trabalhos
with excellent
e
quality of
duas
work
novas tecnologias, né? Minha
and
pergunta vai para a
different
doutora Natália.
technologies. My
E quanto
question
à
is to
parte do
Alexandre. It's
PCR
very
na
important to have PCR in
eu sou muito
the
curiosa,
field
professor, porque eu
at
até
the
te
point of
pergunto,
care.
porque sempre
But
foi
I
a
was
área
very
de
curious
saúde.
about the asymptomatic
Você
cases,
avaliou,
which
você
is a great
pensa
problem to us.
fosse rápido
Uh,
e
you
a tecnologia.
evaluated the rapid
Você
test,
encontrou que
the
na
blood
nova
smear,
tecnologia
and the new
12%,
technology. And you found
comparado a nível, quando você
12% in
colocou,
the new
quando você colocou no
technology
PCR rápido,
compared
PCR
to
rápido, tipo
4% of the rapid test. And the
PCR,
rapid test
PCR
that we
rápido, você
use in
pode
Brazil
durar até 30 dias no
will
sangue.
last
E qual é o resultado
up
que
to
você
30
obteve?
days, the
Porque
blood.
você me falou
And
de alguma suspeita.
the
Será que
the
nesse nível, ao nível
PCR
de
result, it was
persistência, vão ter
similar to
resultados
smear
mais
blood.
rápidos?
But is that because we have resistance of
Então, a
the
pergunta é,
parasite in the
vocês como
rapid
passou
test?
por
The question
aquele
would
teste de
be,
laboratório para saber
have
qual
you
é o resultado?
compared with the qPCR in the lab to know the
Comparou só
results?
com teste
We
rápido.
didn't compare with the rapid
Foi só
test. It
com
was
a
only with
coleta
the
de RT e
microscopy,
o comercial, mas
with the thick blood smear.
comercial não. Comercial foi
But the commercial one
kits de extração
was a
comercial de
kit
PCR
of
de
commercial
DNA e o
extraction of
PCR
PCR, DNA,
comercial
and the
genérico mesmo.
commercial
Então,
PCR
o kit
that is
comercial que
available.
eu
The
comparei
commercial
foi
kit that I
tudo
compared with
molecular
was
no
to
laboratório.
do everything as molecular in the lab. That was the
Quando ele se
approach.
refere ao kit comercial, é um kit
When he
comercial
talks
para
about the
molecular,
commercial
não
kit,
é
it's a
o
commercial kit
teste
for
rápido, que é
molecular.
o
It's not the
TST, esse que
rapid
a gente usa
test
normalmente. Mas eu posso
PSVV
abordar
that we use.
diretamente, então eu vou responder
So
para
I'm
Marta
going to answer
por
Marta
causa do teste rápido.
about
A
the
gente usa
rapid
no
test because
laboratório
we
diariamente
do
com
it in a
pacientes
laboratory every day since
e
we are
paciente
in the
volta
Amazon region and the
três a
patient
quatro
goes
vezes
back for
sem
follow-up. We have
reavaliação.
the follow-up without
Então, o teste
reintroduction
rápido
of new
para
infections.
viva, o
The
parasita tem
rapid
que estar
test
vivo,
for VVEX,
porque a
the parasite has
lactobacillus
to
rhamnosus
be
é uma
alive because the
lactobacillus não existe.
lactobacillus
Porque não
is
existe
a
essa
metabolic
proteína
enzyme. And there
TH2-HR2
could be for
específica. A gente tem na
falciparum. We can
membrana
have it for
dos
falciparum
parasitas vivos,
because
não.
it expresses in the
Isso porque
membrane regardless of the parasite being alive or not. So we saw that in average, we know that the patients don't follow up
estou no
at the
máximo até o
correct
décimo
date.
segundo
So
dia,
we
depois
detected up
nunca
to
mais.
the
Essa é a
12th
posição.
day,
Desculpa,
and then it's never positive
Pedro,
again.
por favor.
Next question. Thank you to the three speakers. Excellent panel. I would like to ask Alexandre if he tested, did you test or compare with other protocols of extraction to see what you are missing using this FTA, if there's anything missing in sensitivity? If you considered, by the figures that I checked, it seems like you are missing something in VVEX, which is normal because usually the parasitemia is lower. So I was wondering if you worked on other targets, other molecular targets to see if you can increase sensitivity. And for costs, I would like to ask Delio as well, the cost per sample, because I remember that it is more expensive. FTA is more expensive. The filter is more expensive. So just to have an idea of the costs of the test. We did not test other protocols of detection. All of the results that I showed are for the gelify reactions. If I have the blood and I test in a laboratory with a standard liquid reaction, I don't see this difference. So I know that part of the loss of results that I had is a problem of my molecular reaction of VVEX that loses sensitivity when it is jellified. And I will draw attention again to the main point of the work, which is not exactly the molecular test. It is only a tool to show that the portable extraction works. The biggest difference, or what we are trying to achieve that we didn't achieve before, is the portable extraction for the PCR in the field. So I know that there's some loss, there's something missing. We identified where it comes from, but I'm not concerned with it because it is not the focus of the work, of the paper. Regarding costs, it is not a pre-screening test for mass testing. It is an active search, so an active case finding. So the cost depends on the amount of reactions that we run. In the best scenario, considering all possibilities, we're in the same stage for tuberculosis and Hansen's disease. So thinking about a scenario where the Ministry of Health would buy 1 million chips, it is about 100 reais per sample. For a smaller number of samples, of course, the cost is going to be more expensive because of scalability.
Ok. Ela pediu, Fátima, sobre o valor, o custo também lá do.
Fatima, she asked about the price of the CBC. It is difficult to find, Thais. People from the companies, they don't give us the cost. They negotiate with you, saying depends on the volume. So it's very difficult to find the specific price. But trying to search in other companies, it would be about $3 to $10. So it is completely inviable using a CBC that would be specific for malaria, the ones with the higher technology. But any family clinic that you go, you have an automated CBC, and maybe the software will be the pathway for us. In terms of costs, they are completely out of touch with reality, I think, the highest levels of technology. Good morning, everyone. I would like to congratulate the panel. Excellent presentations. I am a practitioner, so I also analyze febrile syndromes. We know that from 25 to 50% of the cases of febrile syndromes, we cannot find the diagnosis to them. And you talked a lot about the decision-making power of these tools. So I would like to hear from you about the applicability of these tools, the true applicability in practice, how we can advance in applicability in a scenario where we have more and more emerging viruses and where we miss some other emerging viruses that impact public health. I will start because maybe mine is easier to answer. I was very excited, Tanya, not with what we found now, but with the possibility of what we can find in the future, the evolution of artificial intelligence for data management, which sometimes we cannot analyze because there's a lot of data. We weren't having outbreaks of dengue at the time. In Porto Velho, we had 3 to 4 years of dengue outbreak. We didn't have at the time because most of the population was immunized. But the idea is that this equipment develops other assessments using alterations and morphological factors for practitioners. That might be one of the biggest issues with this equipment, but the question is, what is the best scenario? This is the question by Siemens and other companies. Where would that fit? But I imagine, for example, in a 24-hour family clinic that welcomes febrile patients that are undergoing CBC, the CBC may not detect malaria or give symptoms to malaria. This person may undergo a more sensitive specific test for malaria. The negative is the most common thing for us. We ask the best test that there is, but the patient has to return. They have to go back the next day and the next day, and then there's a bigger chance of detecting it with CBC. I think the best test is seeing the patient again. In the case of the test that I showed, the most important, and that I was happy to see as well, is that it seems that there is a general understanding that we're no longer going to have a general solution that will solve everything. We will need to territorialize and individualize the actions. It was shown here the behavior of the mosquitoes earlier. So I see the test as a new tool to our toolbox. We could choose different tools for different situations. So it is about having these tools available. As not a malariologist, but as a technician, I see these options as tools. I'll speak briefly because I think my colleagues answered the question, but this toolbox is certainly necessary, and we should think about how to organize it and define health policies in the sense so that we can have readiness as well, so that we can predict the future scenarios and have these tools pre-programmed and pre-structured. This screening that we are proposing with photodiagnosis is to attempt to mitigate the need to go to the clinic. In a scenario where we have a center of reference, but we have municipalities in the Amazon that are very remote access. We have trouble commuting in this area. One of the photos that I showed was Eiras do Pará, a municipality in the state of Pará, and for us to go from Belém to this municipality, it takes us 8 to 12 hours. We take a ferry, we take a car, we take a boat, and inside the municipality, we have other complex issues in commuting. The access to doctors is very sporadic. I also showed a photo of another location where we were. I took that photo inside the house of a riverside family that contains a microscope. This person leaves in a canoe and goes from house to house to do an active case finding. These are very valuable services. This is the reality that we have, and I believe we will have for many years. So we need to have this toolbox for these different environments and structure it so that there could be a coherent flow and so that we can have this assertive dialogue with states and municipalities and have very clearly, which I think can be a big challenge,
a gente não está competindo entre a gente.
that we are not competing with one another. We are here helping one another. We're all one great team. We have to have it clear, and we have to have a clear communication so that we can answer this need. It is true, the techniques are not excluding, they are complementary. I'm going to ask one last question to Delio. It is very interesting because sometimes we have a family clinic that does not have a cytometer. You have equipment for CBC only. My question is, with the lessons learned from the cytometer, can you adapt the equipment that you have for an informative CBC for a diagnosis of malaria, or is this impossible? Fatima, this is exactly the suggestion, the challenge that Gates posed to Siemens. And this is a challenge maybe for other colleagues who like informatics or data analysis. We want to prepare something that can be adapted and can update the software of any automated CBC equipment. In order to make this viable in all locations, not only in Brazil, but Asia and Africa as well, it needs to be the simplest equipment possible. Very few channels, not 5 or 6 channels, but the simpler ones. The current algorithms are very fixed. They look at specific points. But this equipment is more malleable. It can alternate positions. I'm an enthusiast of informatics, so maybe all of these promises where people are spending billions and billions of dollars putting together the data centers that will revolutionize health, maybe we should take a step back and use more simple equipment for malaria. Thank you again to the 3 speakers. We leave here with a few very important reflections in this joint search for solutions for different scenarios, especially in areas with active transmission and without transmission. And we should understand how we could take these products to these areas, thinking of eliminating possibilities of reintroduction and maintaining this status sustainably. So once again, thank you very much on behalf of the organizers of the event, and thank you to the audience for the attention to this other panel that we just had. Thank you.